I will get started. Hi, everybody. Thanks for listening to my talk. My name's Jason. I graduated last year from the Gordon Lab at UC Davis, working with Tom Gordon, whom some of you may have met. I will roll right ahead. So as we've heard, Fusarium circinatum is a fungus that causes diseases on pine trees. But one aspect of its life that is less talked about is the fact that it can infect grasses and it produces no disease symptom at all. Many papers have come out in the last 10, 15 years looking at this, showing that it can grow in grasses next to pine plantations in South Africa. It can infect grasses around pines growing in California. And in laboratory environments, you can infect grasses such as corn with this pathogen. So we know that it can infect grasses. And we wanted to look at this in the field. So I did a study in conjunction with Tom and also with the National Park Service out at Point Reyes National Seashore. And we were looking to see if the grasses there are infected. Out at Point Reyes, you see a lot of bishop pine that are infected. You'll see dead trees. You also see branch dieback symptoms, so green branches with scattered brown flag branches in them from cankers. And they get these really large cankers on the branches that can eventually girdle the branch or the whole stem, killing the tree. But in and amongst all of this, there are grasses growing all around the bases of these trees. And we were curious to see if these grasses harbored infections of the fungus. So out at Point Reyes, we collected, with a permit from the national parks, we collected bishop pine tissue, so cankers. And where we collected around them grasses, and we ended up sampling 2 different species of grass. We did Bromus carinatus, which is a native California grass called California brome. Holcus linatus is an introduced European grass called velvet grass. It's very common around California. So we collected these samples over the course of the summer, over several sampling periods in 2016. And then we identified the grasses at the herbarium at UC Davis. So huge thanks to the herbarium and the Center for Plant Diversity who helped us identify the grasses. Then we took them back to the lab, washed them in a series of ethanol and bleach so we could clean the outer surface, and we cultured these plant segments on a medium that was selective for Fusarium. We got isolates from this, and then we wanted to make sure that we actually had recovered the pathogen. So we confirmed that using 3 different criteria: morphology, somatic compatibility, and the ability to cause disease. So I wanted to go through each of those. Morphology was confirmed by looking at the spores under a microscope. What we were looking for specifically, for any fungus nerds out there, was false heads of spores, so multiple spores being held together in a packet. And then these are born, you have multiple false heads being born from a single cell called a polyphyllide. That is characteristic of this species, and other species of Fusarium may not produce this structure. So it helps reduce the candidate pool. So if you see this morphology, that's a good sign that you've got the fungus that you're looking for. Additionally, we tested by somatic compatibility. Essentially, this uses a self-recognition system of the fungus. So if you could imagine a colony growing around an obstacle, like a rock, when the hyphae reach the other end, it is advantageous for those hyphae to fuse if they're from the same colony, because then it allows more efficient transfer of nutrients. However, you don't want to fuse with just any hypha that you come along. So what the fungus will do is it's selective. It only will fuse with or be compatible with other hyphae that are very genetically similar. So what we do is we mutate the fungi to lose the ability to consume nitrate as a nitrogen source. And we grow them on a medium that only has nitrate. So on this Petri plate, we have 2 colonies, one at the top and one on the bottom, and we look at where they meet. If the hyphae don't fuse, there's a uniform structure of hyphae across, and this is a negative reaction. The hyphae didn't fuse. So they didn't recognize each other. That suggests that they're not super genetically similar. However, if there's a positive reaction, the 2 mutants will fuse hyphae, and you get a line of fluffy hyphae of fungus that have regained the capacity to consume nitrate. So if there is a positive reaction, that tells us that those 2 different colonies recognized each other as being similar, and they fused their hyphae together, and that resulted in the visual difference on the plate. What we found is that our isolates recovered from all 3 host species, paired with known tester strains that were known to be Fusarium circinatum. So that gave us a very high degree of confidence that we'd recovered the fungus and not something else. Additionally, somatic compatibility is a more specific distinction than the species level. So there are different somatic compatibility groupings within the species of circinatum. So this was a more granular than just species by itself. So that gave us very high confidence that we had recovered Fusarium circinatum and not a different fungus that happened to also be present growing in the plant tissue. Lastly, we wanted to test if they can cause disease. So we inoculated greenhouse-grown Pinus radiata, Monterey pine, with spore suspensions. And so after that, you look to see if they get sick after several weeks and see if they cause disease symptoms. When we did this, so here I have 5 trees showing you guys. We have a negative control, a positive control, and then 1 representative tree from each of those field isolates we got. So we have a water control, a known isolate from California that had been recovered from Point Reyes as our positive control, and then the field samples. After 1 week, you can see that all the trees look basically the same. So there's not a lot of symptoms. But after 3 weeks, we started seeing yellowing of needles and browning of needles around the point of inoculation. So we saw that in our field isolates as well as the positive control. And by 5 weeks, we found that our trees were looking pretty bad. It seemed that the positive control and all the field isolates that we'd recovered were capable of causing disease, while the negative control was fine. It had no yellowing of the needles. So we observed disease symptoms in both the positive control and all our field candidate isolates, but none in the healthy negative control. So that told us that we had recovered a disease-causing fungus from the grass. This leads to another question, though, which is, if this fungus can get into a grass, is that a dead end? Or is it capable of moving and jumping to another host? In other words, can it produce spores on the grass? To do that, we wanted to test that with the native grass, which grows out of Point Reyes. So we grew Bromus carinatus grasses in a lab, and we grew them in a sterile environment in a growth chamber, and so they wouldn't be exposed to the fungus. After they'd grown to the point where they'd started having senescence in their leaves, we then made spore suspensions and we sprayed them. So we sprayed them all over and then bagged them up for 2 days so it was nice and humid to promote infection by the fungus. We did the same thing with a solution that was absent of spores, with just a water control. So we did this experiment twice, and so we sprayed our plants and then we put them back in the growth chamber to grow. And then at periods thereafter, we would remove senescent leaves and incubate them in a moist environment to see what fungi would grow on the surface of the leaves. So here's our negative control leaves. These are magnified. So we've got clean leaves that we could see, but we also saw some fungi growing off of them. And these fungi were really gorgeous, but they didn't look like Fusarium circinatum. So they had different structures of spores that we had not observed in the picture that I showed you earlier of the one characteristic of circinatum. But I think they're beautiful fungi, so I wanted to show you guys a few pictures. However, on the positive control, or the ones that were inoculated, we saw colonies growing out that were very different. We saw fluffy white colonies, and when we zoomed in into high magnification, we saw that there were spore structures characteristic of circinatum. They look like false heads that told us that it looked to us like this fungus was growing out of the surface of the leaves and producing spores that could be airborne. To further confirm this, we had inoculated these plants with a mutant that expresses green fluorescent protein, which causes it that when exposed to UV light, these spores and the hyphae will glow green, which is not common in fungi because this protein comes from a jellyfish. It's a transgenic mutant. So we took the spores off those leaves and we put them under a scope to see did they glow, and they did. So that told us that the spores we were seeing growing on the leaves of the native grass were indeed the fungi that we had inoculated with and not something else that happened to be present in the experiment. So you can see these glowing spores. Those are the conidia from the fungus. So in total then, this research that we did out at Point Reyes taught us a couple of things. First, Fusarium circinatum infects both native and introduced grass species at Point Reyes. We know that it can infect the velvet grass and the California brome. We're unsure if it infects other species because they were not sampled. But for the 2 grass species that we looked at, both of them were positive for infections of this fungus. Second, we know that what we isolated from the grasses, if you make a suspension of it and inoculate a pine with it, those pines become diseased. So these grass isolates are capable of causing disease to pine. And lastly, we found that if we inoculate the native California grasses with this fungus, those tissues of grass can support sporulation of the fungus under a moist condition. It was in a controlled laboratory experiment, so I can't say for sure that this is happening in the field, but in the experimental environment that we had, the leaves supported the sporulation of the fungus through repeated experiments. I want to acknowledge a lot of people that made this possible. First off, I got to thank Tom Gordon. He was an amazing PI. I really enjoyed researching in his lab. We had a great time. And all these other labs that helped me out, as well as other folks at the university, I have to thank as well National Park Service for letting us sample there, as well as our funding sources. And with that, I'm happy to take any questions.